So we've just talked about transcription and mRNA processing. So once this mRNA leaves the nucleus, it has to go through translation. So translation happens at the organelle ribosomes. Okay, so ribosome is made out of two subunits, a large subunit and a small subunit. And mRNA is read by codons. So right here is the mRNA, which are 3 sequences of nucleotide bases. And the tRNA is this here, which reads these codons with complementary bases called anticodons. Each tRNA has its own specific amino acid attached to it. So, translation also comes in 3 steps. So the first step is initiation. Initiation is the start codon, which is just a sequence of nucleotide bases saying, start here. This is the beginning of the recipe or the beginning of the genetic code for the protein. So the tRNA attaches at this P site. And that's the start, that's the initiation of translation. The second step is elongation. So elongation is the moving of and the reading of, the translating of mRNA and forming each amino acid together via peptide bonds. So what happens is if we pan over again, the first tRNA goes in the P site. The next tRNA comes in through the A site, binds these amino acids together, and then they shift down. The P goes to the E, the A goes to the P, and they keep going on, like we can see here. P is the first one to start, A comes in, E site leaves without amino acid because they're formed together. The third step is once it comes to the end of their genetic code or their recipe to make this protein, it's going to have a release factor saying this is the end. And once that release factor hits the A site, the P site will release this polypeptide chain, which is these amino acids being chained together via peptide bonds. And the result of translation is this polypeptide that has to be further folded and modified in post-translational modification.